welcome to Key Area 5 of Unit 1 of National 5, and that is genetic engineering. So we're going to be having a look at this process and then looking at the specific steps of genetically engineering a bacterium, but also a quick overview of why it's useful for humans and possible crops and that sort of thing.
Okay, so again, this is from the SQA, this is course specification, the things that you need to know about. You can pause it and read it, but we're going to go through all this information anyway. Okay, so what's going to come up in this PowerPoint, we are going to talk about the process of the transfer of genetic information. We're going to look at the actual individual steps of genetic engineering. We're going to look at the role of enzymes in genetic engineering. If you can't remember what an enzyme is, go back and watch the last video about what enzymes are, because it's going to help, because that's the really crucial bit in this. And we're going to discuss what a genetically modified organism actually is.
Okay, so we'll be having a look at the process and why it's useful to humans in order to use it. Things you need to be able to do, so a little bit more specific. You need to be able to name the process used to transfer genetic information. We have already said it about 8 times. See if you can find out what it is. Describe the main steps of genetic engineering. 9. Describe the role of the plasmid during genetic engineering. 10. Describe the role of 2 of the enzymes in genetic engineering. Give an example of real-life use for genetic engineering and explain what is meant by genetically modified organism. So these are things that we're going to have a look at.
Okay, so just for a recap, this is from the very first key area in this, all about your bacterial cell structure. Again, if you're forgetting cell structure, go back and look at that. But a quick recap, these are the structures in a bacterial cell. You have your standard things of cell wall, ribosome, cell membrane, cytoplasm. But the 2 new ones that are very specific to bacterial cells are the plasmids, which are those little rings floating about, and the large circular DNA, which is the big kind of blob in the middle. But the thing we're really going to be looking at in this is the plasmid.
Okay, so just to remind you, genetic information is the code carried by DNA to make proteins. Have a look at the protein synthesis video if you've forgotten this. And proteins are made up of chains of amino acids. So a DNA code should be able to make any protein. Grab that code and put it anywhere, give it some ribosomes and some amino acids, and it should be able to build a protein.
So what actually is genetic engineering? We've said it enough now, it is that process that we're going to talk about. So it is the process in which genetic information is taken out of one cell, so it's extracted from one and it is transferred into a new cell to produce a new protein. So usually taking an organism that does do one thing, taking its genetic information to do that trait and putting it into something that doesn't necessarily do that trait. It will make more sense as we go on though, I think.
So why do we do it? So say we find something that's got a really desirable characteristic and we think, oh, it would be actually really useful if a tomato could do that. Then what we would like to be able to do is take the DNA from the thing that has got the desirable protein and put it into the new organism, such as the tomato. And we can give more desirable characteristics. And this means we can genetically engineer food to improve things like food quality and shelf life and size. So say we find, looking at this example with the tomatoes, maybe we find another plant that has got a really obvious growth gene, okay? And we can isolate this gene, and that's the one that says grow big. And we can take that gene and put it into tomatoes, and then that tomato can make the protein that says grow big. And what we get out of that is bigger tomatoes. Now, there's a little bit of controversy about this whole process. Some people don't like it for possible health reasons. There's none that's been scientifically proven yet that genetically engineered food is bad for health. Other people don't like it for environmental reasons. The idea of genetically engineered pollen spurting about in the air, mixing with non-genetically engineered pollen. And there are some religious groups as well that have an issue with genetic engineering because it's tampering with the building blocks of life. It's worth being aware of these. You don't have to outline them very specifically, but being aware that some people might have a problem with this is a good plan.
Okay, so there are a couple of examples, or there are lots of examples of genetic engineering, but the one that you need to know at National 5 that is going to come up that will be mentioned is the example of insulin. Now in the second unit in multicellular, you will look at insulin and what it does and the role it has in diabetes much more. But for now, it is a really good example of genetic engineering. So if you don't know, insulin is a hormone that usually humans naturally make, other animals obviously make it as well, but people who are diabetic don't make it themselves. So people who are type 1 diabetic don't make insulin. And insulin has a job in controlling your blood glucose levels. So if your blood glucose levels go too high, insulin helps fix that fact. It means that you don't end up having too much blood glucose. But people who are diabetic, they can't do this. And insulin can actually be produced through genetic engineering. It's not always been made through genetic engineering. It used to be taken from pig
and cow pancreases. They
used to be
kill
taken
them and take the pancreases.
But then obviously a lot of ethical issues. People, especially people who are like vegan or certain religions, not very keen on taking insulin out of a pig. But it can now be made through genetically engineering. And the idea is that they are taking a cell that can produce insulin from one organism, extracting that gene.
Oh, from a human, actually. Just a human person who can make insulin.
Just from a human now, yeah. So it might be that me, who's someone who's not diabetic, they would take my insulin producing gene, they would take it out, they would extract it, and then through the process of genetic engineering, they would produce more of this insulin using the process that we're going to discuss. But the idea is they can then mass produce this insulin, and that is the insulin that the diabetic people can then use as their medication.
It's advantageous because, as you said before, like people with vegetarian and vegan issues, not happy about using an animal product as was used beforehand, but also the insulin that they are injecting into themselves is from a human, so it's human insulin. Pig and cow insulin is going to be very slightly genetically different. If you're diabetic yourself, you'll know, but there are different brands of insulin, and they are essentially genetically engineered from different people. And some people find that actually one brand of insulin works a lot better for them than another brand, and that's probably because they're a closer genetic match to the person that they took the insulin from originally compared to the other brand, which had a different person with a different insulin gene. So even human insulin, there are slight differences between humans and what works in terms of insulin. But again, this mass production idea, loads and loads of people, more people, are being diagnosed with type 1 diabetes. That would mean if we didn't have genetic engineering, we would have to be raising an awful lot more pigs to kill spontaneously so that these people could have their insulin to inject. Genetic engineering is a way around that, so it's really good. Another example that we have been looking at is the mega salmon. So it's a salmon that's been genetically engineered to produce lots of growth hormones. So we've taken growth hormones from other organisms, transplanted them into a salmon, and that results in a much larger growing salmon. Okay, and that's better for us because it means a bigger food supply. It means that you don't have to raise quite so many salmon. We don't know the impact that it has on taste or flavor or quality or disease susceptibility of the salmon, but it does produce a bigger salmon, which is better for farmers.
Yeah. Now you don't need to know that this example. You also don't need to know the next example. Insulin is the only one you really need to know. These ones are just a bit interesting and a bit different.
This one here is eternally disappointing. That picture just gives such a cool impression of it. It's a lot less cool than it says. So spider goat is another example. So they took the gene from spiders that said make your web protein, so the silk that they make their webs with, and they implanted it into an embryo of a goat. And what you get is a goat that can produce spider silk in its milk. It cannot shoot webs out of its hooves as shown in the picture. It's a very disappointing thing. But it's in the milk. And then the idea is that the spider silk, we know it's a very, very strong fiber. It can be used for various applications like medical stitches and that sort of thing because it's biodegradable and really, really strong.
So on to the actual process of genetic engineering, what is actually happening. Now, before we even start this, the most crucial thing is your wording has to be bang on in this. It was the extended answer question. It was 4 or 5 marks 2 years ago. And it was harsh. They were very, very strict, and you would get penalised if you used the wrong language throughout. So just make sure that this one, you are learning it and you are learning the definitions exactly as we're going through them, and you will be fine.
Now, it's crucial. We don't cover genetically engineering, say, a goat or a tomato. Instead, what we look at is taking a bacterium, a really simple cell, and genetically engineering it to produce, say, a useful human protein like insulin. Okay, so stages 1 and 2 are seen in the diagram in front of us, but the idea is step 1, the correct chromosome with the required gene is identified. So for example, you want to produce insulin. What you do is you take a healthy human, you extract a cell from that healthy human, and you look inside their nucleus. In their nucleus is every single gene that they have. In every nucleus of every human, me, you, everybody, is every single gene that you have. Okay, and what you do is you find the right chromosome that's got the right gene on it, And then what you have to do is you have to cut that gene out of that chromosome. Each chromosome can contain hundreds, thousands of genes. You only want one. So you need to identify the gene that you want, and you need to cut it out of the chromosome. So what we've got is a tiny piece of DNA code that says build insulin. That's what we're left with at the end of this stage. Okay.
Okay, the next thing, so once that gene has actually been extracted, and this doesn't, it almost happens hand in hand with the first stage because it's almost beside it,
the same time.
is that you will get a plasmid. So from a bacterial cell, you will take a plasmid and it will be extracted from that bacterial cell. And then the plasmid, and this is where it's really important, is cut open using an enzyme. So in the success criteria for this, we said that you have to be able to say when enzymes are used for genetic engineering. This is the first time. There's only 2 times we will say them. This is the first time. So the plasmid is cut open using an enzyme. You don't need to name the enzyme. You don't need to know anything about it other than it is extracted from the cell. It is then cut open using an enzyme.
That word open is super important. If you say plasmid is cut using an enzyme, cut open, yes. Okay, they really don't like cut because they're like, oh, do you mean like cut in half, cut in quarters? No, it's cut open. It's a ring and it's cut open to make a ring with a gap in it. Okay. Into stage 4, we've got our extracted gene on the left, our cut open plasmid on the right, and they are joined together. Okay, so you put the extracted gene into the plasmid, which is again a ring of DNA. So you're just joining human DNA onto bacterial DNA, and that is sealed using a different enzyme compared to the cutting one. Instead, we're using basically a gluing enzyme. Okay, now the different enzyme is underlined because you need to be able to specify it's a different enzyme from the cutting open one. Okay, that's why it's underlined.
Okay, once that has happened, the plasmid with that new gene now sealed into it is then placed into a new host bacterial cell. So the plasmid with the gene in it, it's not put back into that original bacterial cell. There is a new cell that has been brought in and it has been placed into that. And that's really important that you show that you know it is a new host bacterial cell that this plasmid is inserted back into.
And stage 6, these bacterial cells can reproduce. What they do is they divide and make more copies of themselves. So they split in 2, and then those 2 that are made can split in 2, and so on and so forth. And you can make lots and lots and lots of those genetically changed bacteria, and each of those bacteria can now make the desired protein. In this case, let's say it's insulin. So you get a bunch of bacteria that have the ability, is when you can extract off the fluid on the side of them, is that you can extract off insulin, and that is really, really convenient and really, really handy, considering it's human insulin from a human person, but it's put into a bacterium, and you're just farming these bacteria for yourself. Okay, and that's the end stage. Now, if we have a look at an overview, you might want to pause this so you can have a little bit of a closer look, but this is how the process looks when you combine it all together. So up the top, we've got our human cell with the chromosome that we want being identified, and then we cut the gene that we want out. The bacterial cell with the plasmid's pulled out and cut open, and then the idea is your new gene and your plasmid are joined together, and then we've got our plasmid back into a different host cell, so a different bacterial cell, probably destroyed the first bacterial cell, removing its plasmids. Okay, so we pop it back into a new host cell. That host cell divides and divides and divides, and it produces your desired protein, and in this case, it is insulin.
Okay, so just to highlight the difference of this, that is the process of genetic engineering. Something that has been genetically engineered, we would then say it is a genetically modified organism. So when you hear things talking about GMO, they talk about it a lot in food produce now. Something that is GMO means it is a genetically modified organism, and that's something that some people are really, really not keen on. They don't like GMO stuff, and that is because they don't like the idea of it's had its genetics played with, and it's not natural, so to speak. But that's what a genetically modified organism is, just simply one that's had its DNA modified through the process of genetic engineering, so it now contains a new gene that, if it was to grow naturally, it wouldn't necessarily contain.
So we can do that with food, or in the previous example, that was the bacterium that had the insulin gene. That bacterium is a genetically modified organism. Now, reasons why people lose marks in this whole process. For one thing, talking about the fact that the required gene or plasmid is extracted using enzymes. That is not correct. The enzyme is just, sorry, not the enzyme. The plasmid is basically grabbed, let's say with a pair of tweezers, and pulled out of the original bacterial cell. It's not really tweezers, it's a little bit more complicated than that, but it's definitely not enzymes. And then saying that the chromosome is extracted using enzymes, no. Again, it's just pulled out, okay? Not stating that the required gene is extracted from a chromosome. That's another way that people lose marks. They forget that the required gene needs cut out of its original chromosome. Remember, a chromosome might have, 3,000 genes on it. You only want one of them. You have to cut that desired gene out. This is the most common one. People use too basic language. So things like the plasmid is taken out of the bacterial cell. That's not good enough with your language. The gene is put into the plasmid. No, it's sealed into the plasmid. You need to be 100% accurate with this language. And then the final reason why people lose points is they state that the plasmid is inserted back into the original bacterial cell. No, it is not. It's put back into a host cell, or it's put into a host cell. Okay, so a different cell than when it came from, it's shoved into a new cell.
Okay, so just to summarise genetic engineering again, so genetic engineering, it is the artificial transfer of DNA from one organism into another.
Okay, the source cell is the cell that contains a desirable gene from a human, from a thing that's got a desirable characteristic.
The plasmid, that's that little ring of DNA that's found in a bacterial cell. These are extracted and then they are modified during genetic engineering.
Okay, the bacterial host cell is the cell that the genetically modified plasmid, the newly changed one, is put into.
And insulin, that is that really common example. It's the one that's going to come up. It is the protein that can be produced using the process of genetic engineering.
And that's it. That's the whole of that key area. Done. Okay, so you can see how short it is. The reason why it's so short is there's a few points of very accurate language that you have to be able to get, but your process used to transfer genetic information, hopefully you got it, is genetic engineering. The main steps of genetic engineering, remembering to be accurate about plasmid being cut open, the two places where you see enzymes, and then stating that it's a different enzyme that seals in the new gene. The plasmid is going to be the thing that's extracted from a bacterial cell, cut open, new DNA added in, and then put into a new host cell. Do you want to cover last three?
The role of the two enzymes, so you've got to be specific with these. It is the ones that are going to cut open the plasmid, and we've got the different one that is going to seal that required gene back into the plasmid. An example of a real-life use for genetic engineering, so that is the production of insulin, and explaining what is meant by a genetically modified organism. That is simply an organism that's had its genes tampered with or changed in a way so they are not producing what they naturally would produce.
And that's it. So the next key area is respiration, and that'll be the final key area of Unit 1. Nearly there.